Welcome. Today we are going to learn about the response elements. Response elements are the cis-acting nucleotide sequences. What they are the cis-acting nucleotide sequences to which the regulatory transcription factor it can bind. That is, this cis-acting nucleotide sequence, it is going to be recognized by the specific regulatory transcription factor, thereby causing the gene response to various regulatory agents. Okay, so the response element, it is a DNA sequence, okay, where the true activator it is going to bind. Where it is located, this response element, it may be located into the promoter, into the enhancer, or it may be 200 base pair from the start point of the gene. And when you look at the response element, you can find that it has a diode symmetry. Okay, that is it. When you read from 3' to 5' or 5' to 3', the sequence it is the same one, that it is a diode symmetry. And to such a sequences, the proteins or the activators which are going to bound that activators, they are the dimeric one. Okay, now there are number of examples of these response elements. Some they are summarized here. Okay, first one, it is a heat shock response element. Okay, to which the HSTF, this inducible factor or this activator, it is going to bound, and this response element, it may be present into the HSP70, HSP83, HSP27. Okay, then the CRE, it is going to present into the somatostatin, fibronectin, CFOS, HSP70, alpha gonadotropin. That is about the in the 5 genes, this CRA response element it is present, and its activator it is a CREB and ATF. Okay, then there is a 4-ester response element, okay, which is present into the metallothionein 2, alpha-1 antitrypsin, retinoic acid response element, whose inducible factor it is a retinoic acid, and the response element it is present to the growth hormone, myosin heavy chain, then the glucocorticoid response element, GRE, then there is a serum response element. Now when you look at, these response elements, they are present in more than one gene. Okay, that is, a gene, it can respond to the number of environmental conditions, and to respond to that number of environmental conditions, there is presence of the response element, so that response element, that particular activator, it is going to bound and it is going to respond to the changing environment. Okay, but when you look at the conscious sequences of this response element, here you find that these conscious sequences, they are going to differ from one another, as the activator which is going to bound to this response element, it is going to change. The conscious sequence, it is also going to change. That is, the response element, it is a particular sequence of DNA. Okay, it is very specific one, and to that specific DNA sequence, okay, a particular activator, a particular inducible factor, it is only going to bound. Here, the example here, it is a metallothionein gene. Okay, it is the gene which is an example of the flexible gene expression control. Okay, metallothionein gene is an example of the transcriptional regulation in eukaryotes. Metallothionein protein. Okay, metallothionein gene, it is going to express the metallothionein protein, which is which works when concentration of the heavy metals is high. It binds to the heavy metals and remove the heavy metal from the cell, thus the protect the cell. Now there are number of heavy metals. Okay, whenever heavy metals enter into the cells, okay, like the silver, lead, or gold, okay, these are the heavy metals, and these heavy metals, when enter into the cell, at that time what is going to happen, that heavy metals, it may bound to the proteins which are present into the cell, and it is going to collapse the metabolism. Okay, so whenever whenever the heavy metals other than the silver, gold, there are the number of heavy metals which can enter into the cell, and when these heavy metals they are entering into the cell, when they are collapsing the metabolism, there is a question over the survival of the cells. Okay, so to survive the cell, that heavy metals, it has to be thrown back into the environment, and for that purpose the protein which is going to come, it is a metallothionein protein. Okay, metallothionein gene, it is going to be expressed at a higher level when there is an incoming of the heavy metals into the cell. So the metallothionein, what it is going to do, it is going to bound with the heavy metals, and it is going to pull that heavy metals outside the, or it is withdraw the heavy metals outside the cell. It will throw the heavy metals into the outer environment. Okay, so when you look at the regulatory elements of the metallothionein gene, you can find out that there are about the 4 response elements they are present: GRE, BLE, MRE, and TRE. Okay, GRE, it is a glucocorticoid response element, BLE basal level response element, MRE, it is a metal response element, and the TRE, it is a phosphorestor response element. Now, when you look at the regulatory elements, you can find out that the GRE, it is present only in 1 number, BLE, it is present into the 2 number, the MRE, it is present into the 4 numbers, 1, 2, 3, and here it is a 4, and the TRE, it is in the 1 number. And when you look at the MRE, it is present into the promoter. This GC and the Tata, these are what? These are the promoter sequences, and here you can find out that the MRE, it is present into the promoter, and some of the MRE, that is a metal response element, they are present outside the promoter. That is, the response element, it may be present into the promoter or it may not present into the promoter. Okay, and here you can see that the number of response elements which are present into a gene, it can vary. It may be 1, it may be 2, it may be 4. Okay, in this way there may be presence of the number of response elements. Accordingly, what level of the expression it is required. Here you can see that MRE, it is present into the 4 numbers. MRE, it is a metal response element. Whenever there is a incoming of the heavy metals into the cell, that heavy metals, it has thrown back immediately. That is whenever there is a incoming, whenever the heavy metals they are entering into the cell, before they are going to collapse the metabolism, that heavy metals, it has to be captured and it has to be thrown back. Okay, so for that purpose, there should be the high level of the expression of the metallothionein gene. The number of the metallothionein protein which is present in the cell, it is to be at a higher level. So there are number of MRE. So to the MRE, who is going to bound? The activator, it is going to bound. When activator it has been bound, there will be the loading of the basal transcription apparatus. That is, there will be what? There will be the formation of the basal transcription apparatus at the at where there is a presence of the activator. Okay, so as the number of activators loaded, they are the more, as the MRE, they are the more, the basal transcription apparatus which is going to be loaded over the gene, it will be at a higher level. So there will be the high level of the expression of the metallothionein gene. Because expression level, it has to be higher one. For the high level of the expression, there should be the high level of the transcription rate. That is, there should be the high efficiency of the transcription. And to have the high efficiency of the transcription, there should be the efficient loading of the RNA polymerases over the promoter, and that it has to be achieved by having the number of response element at a higher number. Like here, it is in the 4 number it is present. Okay, so in this way, metallothionein gene, it has the 4 response element. It is the GRE, BLE, MRE, and the TRE. BLE, it is to maintain the basal level of the metallothionein protein. Okay, GRE, it is allowing the response when there is a glucocorticoid hormone it is present. TRE, it is a phosphorestor response element. Okay, so in this way, it is a metallothionein gene. I think you may have cleared the idea about the response element. Okay. If you like my video, please subscribe my channel and also share my video with your friends so the knowledge it is going to spread. Thank you.